The overall goal of this procedure is to extracellularly identify individual motor neurons for a motor pool, then to monitor and control them during motor patterns. This is accomplished by first placing the prepared buccal mass of Aplysia with hook electrodes attached to key nerves in a specialized recording dish, and then preparing the buccal ganglia and cerebral ganglion for extracellular stimulation and recordings. The second step is to attach suction electrodes to the key nerves and muscles of the buccal mass, to attach force transducers to the target muscle, and to place extracellular glass electrodes onto the sheath of the buccal ganglia. Next, locate the target motor neurons with the extracellular glass electrodes. The final step is to extracellularly stimulate and record target motor neurons while the muscle contraction forces are measured simultaneously. Ultimately, the extracellular stimulation and recording technique is used to show the identification of individual motor neurons and their neural activity during different feeding-like motor patterns.
The main advantage of this technique over existing methods like intracellular identification of neurons is that extracellular glass electrodes can identify and monitor multiple motor neurons in the same preparation during muscle movements. Helping to demonstrate the procedure will be Jeff McManus, another graduate student from my laboratory.
In this procedure, to prepare the suction electrodes, first cut a piece of polyethylene tubing about 10 centimeters long. Hold the tubing on both ends and place it very close to the flame generated by the Bunsen burner while rotating it until it becomes soft. Then stretch the tubing carefully while moving it away from the flame. The middle part of the tubing will become elongated and narrowed as it is pulled.